Hello friends, welcome to my channel. In this video lecture we are going to understand a very important topic, that is the Human Genome Project. Human Genome Project. So today let's understand the project of Human Genome and why it is called the mega project. So first we need to understand these terms. So human means it is related to us, and genome means it is the set of, the total number of genes. It is the total number of genes present in haploid set of chromosome. So in our haploid chromosome, so here it is about human beings. So the total number of genes present in haploid set of chromosome of what? Of human is human genome. So of any organism, it's called the genome of that organism, of any organism. So we need to know that genome means it is the total number of genes present in haploid set of chromosome of any organism. So the haploid set of chromosome present in the total number of gene in haploid set of chromosome in human being is known as the human genome. And the Human Genome Project is a mega project carried out or started by James Watson. It was started by James Watson in the year 1990. Exactly the date was 1st October. And Colin was in charge of the project from 1993. From 1993, Colin was in charge of the mega project, Human Genome Project. And it was started by two organizations, the US Department of Energy. So it was started by US Department of Energy. One is the US Department of Energy and the second one is the National Department of Health and National Department of Health. So these are the two organizations who supported the Human Genome Project, and the fund for this project was supplied by Wellcome Trust of UK. So the Wellcome Trust of UK supplied the fund. The fund was provided by Wellcome Trust of UK, and two organizations responsible for carrying out the mega project are US Department of Energy and National Department of Health. So remember, there are certain countries who also took part in the process of the Human Genome Project. They are Japan, France, Germany, China, etc. So these countries also supported this mega project. So now we will go for the estimate. So what was the estimate for the project? Let's see. So now let's discuss about the estimate. So as it was planned to carried out, so the estimate was, we need to understand what was the estimate before the start of this project. The estimate was that this project will finish within 15 years. So within 15 years this project will be finished. And another thing was that the genes, total number of genes that are going to be discovered may be from 80 to 80,000 to, it was estimated that the total number of genes may vary between 80,000 to 1 lakh total genes. It was estimated. Okay. Another thing is that total number of base pair was estimated to be 3 into 10 to the power 9 base pair. Total number of base pair 3 into 10 to the power 9 base pair. Okay. So total for a single base pair, the cost is approximately, it was estimated that the cost is approximately $3. So for one base pair it will take $3. So $3 for one base pair. So the total number of base pair is 3 into 10 to the power 9. So around 9 billion 9 billion US dollar are required for this mega project. This was the estimate. And another estimate is that if the total database or the total base pairs are stored in books, then if the book has 1000 pages, the book has 1000 pages, one book carrying 1000 pages, that on each page carry 1000 letters, then the total number of books required to store the base pairs in books will require minimum 3300 books which are having 1000 pages, and each page have 1000 letter. It is the estimate of the mega project, but actually it was found that 20 to 25,000 bases are there, not 80,000 to 1 lakh, but it was found at the end of the project that the total number of base pair is 20 to 25,000. 20 to 25k means 1000. Okay, so another thing was that it was estimated to finish in 15 years, but it was finished 2 years before. That means in 13 years it was finished. It was started in the year, I have already told you, it was started in the year 1900. Okay, and it was finished in the year 2003, except a single chromosome. Except one chromosome, all the other 23 chromosomes, the base pairs are already discovered and their roles are already known by 2003. 2 more years are required for the experiment of chromosome number 1. So to carry out the base pairs or the bases or the genes of the chromosome number 1, it took another 2 years, means the project was completed in the year 2006, which took chromosome number 1, and the chromosome number 1 is very important, so that its genes required more 2 years to finish the mega project. So as it took many years and a great amount of cost was involved in that project, it is considered as a mega project. Now let's discuss about the salient features of Human Genome Project. So let's see, here we have the first point. The human genome contains 3164.7 million nucleotide bases. So total number of nucleotide bases is 3164.7 million. Next, the second point, average gene consists of 3000 bases. So the total bases are 3000 in every gene. Average, if you will take, each gene has about 3000 bases. One is there, which is the name of the gene is dystrophin. Dystrophin is the gene with highest or the largest number of bases. So the largest known human gene dystrophin is having. So the dystrophin is having 2.4 million bases, which is highest among all the genes. So remember, dystrophin having 2.4 million bases. All others are having an average basis of 3000. So remember, only the dystrophin is having highest number of bases. Next, 99.9% nucleotide sequence are same. So in every human beings, me, you, and each and every human beings you see around yourself is same to you, 99.9%, because the bases are, nucleotides are same by 99.9%. Only 0.1% difference is there between you and me. Okay, so another point is 50% of gene functions are unknown. Okay, So only 50% of the genes we have discovered their functions, and other 50% are yet unknown. Next point, less than 2% of genomes codes for protein. Only 2% or less than 2% of genomes codes for protein. Next point, 1.4 million locations with single base pair difference. There are 1.4 million locations in our chromosome where there is only a single base pair difference, means SNPs. SNPs means single nucleotide polymorphism. Okay, so what is single nucleotide polymorphism? Means look, here the location is having A, T, G, T, A, T, like this, and another location, in another location there is also A, G, T, A, T, like the first one, and here only the in place of T only there is a G or there is a C. That is the difference. Only a single base pair difference, which is known as single nucleotide polymorphism. Clear? So these are the salient features of Human Genome Project, and these are the outcomes of the Human Genome Projects. Another very important thing we should understand here, the chromosome 1. The chromosome 1 is having maximum number of genes, that is 2968. So 2968 number of genes are present in chromosome 1, which is maximum among all the chromosomes. And the minimum number of genes are found in chromosome Y, and that is 231 or 231 only. So this is very important for your board examination as well as for NEET examinations. Remember, chromosome 1 having maximum number of genes 2968. Chromosome Y having minimum number of genes 231. So these are the important salient features of the Human Genome Project. So please remember these features, these are very important. Okay, now we discuss about the methodologies. So what is the method by which we will do the Human Genome Project? So first we need to understand the DNA. So DNA has two different parts. One is the coding part, another is the non-coding part. This is the non-coding. Okay, so we have two parts, coding part and non-coding part. So let's see what are the two approaches. There are two approaches. So what are the two approaches? They are, first we need to do total DNA sequencing or sequencing of only the coding part. So we can do sequencing of coding part only, okay, and another one is we can do the sequencing of the total or the whole DNA, sequencing of entire DNA. Another is sequencing of entire DNA. So there are two different approaches. So first one, we will see how we can do the sequencing of coding part and what is it. So sequencing of coding part means which is expressed sequence tags, known as EST, expressed sequence tags. Okay, so it is EST, it is known as EST. Okay, so sequencing of coding part only is known as expressed sequence tags, because the coding or sequencing will be done of the expressed part, which expresses something or codes for something. And here in the second case, we will sequence the entire DNA. It is also called as sequence annotation. It is called sequence annotation. Okay, so sequence, here E is there. It It is sequence annotation, sequencing of entire DNA. Sequence annotation. Okay. It is the sequencing of both the part, coding as well as non-coding. Coding plus non-coding. Non-coding means entire DNA. Here the sequencing of coding part only is done. Okay. So this is the two approaches. One is the sequencing of entire DNA, which is known as sequence annotation. Another is sequencing of coding parts only, which is known as ESTs or expressed sequence tags. Now we will go for the process. So what is the process of making or carrying out the process of human genome project? Let's see how we can do human genome. How human genome project is carried out. What is the process? So let's see about process. So in the process we have the DNA. We will cut it by using obviously the biological cutter, the endonuclease, cut with the help of endonuclease, and after the cut we will get various fragments of the DNA. We will get fragments of DNA, and after getting the fragments we will carry out cloning. And for cloning we need host. For cloning we will need host, and the host here are mainly of two types. One is the bacteria, another is the yeast. So remember, the host may be bacteria or yeast. When the host is bacteria, the vector required for the process is bacterial artificial chromosome. It is known as bacterial artificial chromosome. Bacterial artificial chromosome. And when the host is yeast, we will need yeast artificial chromosome. We will need yak or yeast artificial chromosome. Okay, so these are the two hosts in the process of cloning of the DNA fragments, and the host may be bacteria or yeast. When the host is bacteria, we will need the vector, which is the bacterial artificial chromosome, and when the host is yeast, we need the vector yeast artificial chromosome. So these are the two vectors we need. Now let's see how the fragments, after getting the fragments, we clone them by using the vector inside the host body and sequence it by a method known as Sanger method of sequence. The method of sequence is known as Sanger method of sequence. This is the name of the scientist Frederick Sanger. We know him very well. He is also known for the determination of amino acid sequences in protein. So Frederick Sanger is also known because of sequencing the amino acids present in the protein, and this method is also, Sanger method is also used for sequencing of DNA in case of human genome project. Now finally, let's discuss about the applications of the human genome project, where the first point is diagnosis and treatment of diseases. So by understanding the genes which is responsible for causing certain disease, we can make changes to treat the disease or treat particular disease. Next one is the recombinant DNA technology. This technology is used to modify a specific gene that is responsible for any disease. Third one is in the field of agriculture also we can use the genome project. By understanding the genome of a particular plant, we can make hybrid plants by using various technology. Next, the chemistry of life. We can understand the chemistry of our life by understanding various genes present in our body and their role in our body organization. And the last one is in the field of evolution. By this process, by knowing the genome, we can easily know from which organism a specific organism is evolved. So it has also evolutionary importance because it helps in identifying evolutionary relationship between various organisms. So this is all about the estimate of Human Genome Project. What is the real outcome of the Human Genome Project? Who are involved, which organization is involved, who supplied the fund to this project, why it is called mega project, and what are the methodologies, process and applications of Human Genome Project. Thank you very much.